Supernovas, the Tree of Life, and the Human Genome Project. Who would have thought that we could interrelate these three concepts? Well, we've tried, and here we go. Let's begin with a star. So this star is just happily living its life out in space, hydrogen becoming helium and energy, helium becoming carbon and energy, carbon becoming oxygen and energy. And at a certain point, when it's very old, it has a sudden core collapse. In other words, the nuclear fusion occurring in the star can no longer sustain the core against its own gravity, and thus it collapses inward at very high speeds, resulting in extremely high temperatures, and then it expands outward again in a massive explosion. This is called a supernova. But that is not the only way a supernova can be created. There is one other way involving white dwarf stars. When a white dwarf star either manages to accumulate more mass than about 8 times the mass of our Sun, or two white dwarf stars collide, then they cannot sustain their cores and thus also collapse inwards and explode, creating a supernova. During the violent supernova, temperatures are sufficiently high to fuse elements that are not normally formed in stars. This results in the production of many heavy elements, including iron. When the supernova explodes outwards, it ejects these heavier elements, together with lighter elements, including potassium, calcium, carbon, and oxygen, all the organic building blocks for life. We will follow one particular iron atom to Earth. So our iron atom collides with Earth, around the time of the Cambrian explosion, where life is about to flourish. It could easily become part of one of 3 major forms of life: the eukaryotic, archaean, or bacterial. These are the 3 main branches of the Tree of Life. We still don't know when or in what form life first came to be. Each of these forms is similar to the untrained eye, but they have fundamental differences which are key to the diversity of life. Despite being too small to see, bacteria make up the vast majority of biomass on Earth. Bacteria are some of the most diverse and excellent adapters, and can be found in extremely diverse circumstances. Archaea are similar to both bacteria and eukaryotes, but there are important distinctions to make. They are very small and simple organisms, but unlike bacteria, they have no nucleus. Yet, like bacteria, they've remained pretty much morphologically constant throughout time. Finally, eukaryotes make up the 3rd branch in the Tree of Life. These are architecturally by far the most complex life forms, and their emergence remains one of the biggest mysteries in science. Eukaryotes are the most well-known form, as they have evolved into nearly all of what we can see today as life. Plants, animals, and fungi all fall into the eukaryote branch in the Tree of Life. Virtually all life that is visible with the naked eye is in fact a eukaryote. But what does this tell us about ourselves? Centuries of scientists have worked from discovery to discovery to find out what makes us human and how, and if, we are different from animals. To understand how different we are from animals, we can compare something called the genome, the nucleic acids that make up our genetic code, to that of other animals. In the not-too-distant past, an undertaking was completed to codify all 25,000 genes of our DNA, which was called the Human Genome Project. This project took over 20 years to finish, and is often heralded as one of the greatest triumphs of science. How does it work? In a nutshell, our genetic code is made up out of things called chromosomes, which in turn are made up out of wrapped DNA strands, which in turn is made up out of a very long string of genes, which in turn is made up out of base pairs, which you can read with very complicated and difficult procedures that we won't go into right now.